Hi, very good morning everybody. Today we discuss about animal cell culture in this video. Why animal cell culture? In recent days, people dependent on many animal models which is ethically bounded and also time consuming. So people prefer this animal cell culture in vitro model is a preclinical tool where we can get maximum data, and then this can be translated to in vivo model to understand in a best way. So the first cell culture is used by Ross, Harrison and team using frog nerve cells by hanging drop method. The media used then is chick embryo plasma. Even people used whole blood and also embryo extract, but people got a success in different segment, in different percentage of success rate. But over a period of time, people realized this cell culture is indispensable for growing virus, where the importance of vaccine come to existence in the presence or by understanding this cell culture related vaccine production. This particular cell lines are available in different cell repository centers like NCCS, Pune in India, ACC, UK, ATCC, US. To perform this mammalian cell culture or animal cell culture, we need an aseptic condition. Hence, we need to abide with certain rules and regulation where we have to follow certain methods to keep aseptic condition. So in that, some of the methods we discuss: wet heat sterilization, where we use autoclave for sterilization of media and other liquid products. Dry heat sterilization we use for any dry glasswares that you use for sterilization. Filtration, if any liquid media have any heat sensitive chemicals or products, where we can go for membrane filtration technique. Irradiation, where we use UV. Chemical sterilization, where we use ethanol and formaldehyde. The basic question: what is the difference between primary culture and secondary or continuous culture? Normally primary culture are where we take the source directly from the animal tissue. So that will have capacity to grow only for less number of time. Normally those sources will have heterogeneous cell population, and very important, which will have finite lifespan in vitro condition. The same cell in vivo, it will grow any number of time because that is controlled by several factors in the body. But in vitro condition, this will respond only for less division pattern. This will be mainly anchorage dependent and exhibit contact inhibition. Whereas continuous cells or secondary cells are nothing but which is derived from primary culture, or you can grow or produce by spontaneous mutation or by viral transformation. But this set of cells are uniform in nature, which have loss of anchorage dependent, meaning it does not need a substratum to attach, which will express loss of contact inhibition, and more important, indefinite lifespan, meaning it will grow continuously in vitro condition. There is no like a barrier in division pattern or time, number of time division. And very important signature, genetically unstable. Types of cell lines that we have are being used as a cell model: HeLa, HL60, 3T3, COS7, HCHO, HEC293 and endothelial cell HUVEC. How they produce continuous cell lines? By spontaneous mutation method, chemical transformation method, and viral transformation method. Difference between adherent and non-adherent. There are two types of cells. One is adherent cells, other one is suspension cells. So adherent cells, that name implies, which needs attachment, some solid support or semi-solid support. Suspension cells, where the term implies which remain suspended in the media, which will do not attach to the substratum, rather it will go and sit in the surface, where it will divide radially. So both the cells are visualized using a microscope to understand the morphological pattern, like how cell divide. And very important thing is, adherent cell do not need agitation, whereas suspension needs agitation. Likewise, adherent cells need a trypsin immediate to detach, whereas suspension cells do not need that trypsin to detach. So both the cells are used for protein production or any transcription factors production or any special protein production. Common cell culture media that we use: Eagle's minimal essential media, DMEM medium with high glucose and low glucose, RPMI, Lebowitz, Iskov's modified Dulbecker's medium. So these are modified medium of minimal essential media, meaning over the period of time, the minimal essential media become formulated, designed to EMEM. Again, that was modified to become DMEM, and henceforth we get the recently available special medium like this. So RPM is basically used for lymphocytes and most of the cancer cells. DMEM is people consider that as a basic or universally common media that used for any cells. But if any special cells like neural stem cells, where we go with a special media with special growth factors. Sorry. Composition of media. What are the components present in that media, what we call DMEM, EMEM, RPMA? So these are general components present over there. Basal salt. So that salt, inorganic salt, which maintains physiological pH and osmotic pressure in the growing cells. Likewise, the ions like sodium, potassium, magnesium, copper, which is also another important factors which drive cell to divide and cell to communicate. Carbohydrates, it's a basic source for energy to any cells that we grow in vitro condition, where we provide glucose, maltose, sucrose, mannose and fructose. Amino acids, it's one of the small molecules being used in the media preparation, which is used for cell communication, which is responsible for promote cell attachment. So, people observed amino acid deficiency inhibits cell division, also lead to mutation where chromosome damage or disorder occurs. Vitamins, another small molecules which is used as a cofactor in metabolism of cells, and especially choline is necessary for incorporation of membranophospholipase. Protein supplements, there are a lot of growth factors, lot of transcription factors and proteins being used as a small molecules in different event, like transport protein acting as a to bind with iron, which helps in uptake of iron. So there are different small molecules get involved. Antibiotics, normally people don't use antibiotic in cell culture, but if necessary arise or need arise to control the microbes, people use antibiotics. So the normally used antibiotics are penicillin, streptomycin. To control fungi, people use gentamicin and nystatin. Components of sera, there are general composition of sera like growth factors, trace elements, lipids, steroids, attachment factors, transport metabolites and protease inhibitors. Whereas there are the factors how that is correlated with functions, like albumin transport the lipids, fibronectin promotes cell attachment, transferrin bind with iron, insulin uptake of glucose and amino acids, PDGF mitogen for fibroblast, hydrocortisone promote cell attachment. So there are many functions, there are some of the macromolecules or small molecules yet to identify from serum. Therefore, people use in certain cases serum-free media for cell culture. Cell culture contamination is another important segment. Bacterial contamination. This is the first and foremost people have to address. As I said before, antibiotic is not required. Suppose in the laboratory where they encounter repeatedly with contamination, where they have to use antibiotics. So, bacterial contamination is the one which is based on their morphology and pattern, we can easily identify with microscope. Likewise, mycoplasma, but this is also a bacteria, but which lacks cell wall, which cannot be identified using microscope. Yeast contamination, this is another repeated contamination. Very oftenly people encounter those who are doing cell culture. The morphology or contamination can be identified by turbid in nature or change in pH. Easily we can discard the infected flask. Likewise, cross contamination, that's another important thing. If people using a same incubator where with different cell lines being cultured over there, in a single flask you may notice more than one cell line is growing. So, to avoid aseptic condition strictly followed, but very oftenly people has to do a DNA sequencing to avoid this cross contamination. How to detect contamination? So, probably the contamination can be observed like in a turbid culture media like this, change in growth rate, abnormal high pH, poor attachment, multinucleated cells or some of the signature of contamination. Yeast, bacteria, fungi usually show visible effect like change in media and turbidity as a common signature. Mycoplasma is detected by DNA staining using EKIS, which is a fluorochrome. Using a fluorescence microscope, we can detect this contamination of mycoplasma. Likewise, mycoplasma contamination also detected by PCR amplification, where we use mycoplasma RNA. The best way to eliminate contamination, please discard the infected flask or contaminated flask so that we can save the other flask that we use in the same incubator or same cell culture lab. Thank you for watching. We will meet in the next video.